Good morning, everyone. My name is Prasant Mohapatra, and I serve as the Vice Chancellor of Research here at UC Davis. It is truly a delight to be gathering once again in person and to see so many faces. I would like to welcome you all, including our online attendees, for the 2022 Research Expo. We are truly excited to host this event after a 2 years gap because of the obvious reasons you all know. The goal of the Expo is to highlight some of the most exciting research to inspire new ideas and collaborations. And we also want to showcase the resources that are available to help you advance your research programs. And as you all know, the last 2 years have been really difficult for sure, but I would like to express my gratitude and admiration for the way our research community responded to the challenges that we faced. Not only did people do what was asked to support the community from an operational perspective, many of you leaned into the challenges and provided insight and solution that brought us to where we are today. Otherwise, we won't be having an on-site event like this. Others of you adapted to continue and advance your work, showing your commitment to and your passion for innovation and discoveries. Last year, our campus set an all-time record of external research funding of $968 million, which is an amazing feat. And this year, we are on track to beat that record once again. And personally, I'm reasonably confident that we will cross the $1 billion mark of external funding research during the year. Okay, so that will be a unique achievement. This research and related innovation has fueled a growing impact in our world, something everyone in this room should be proud of. I would like to thank you all for your contribution to this impact and hope that today's event will offer you more advantages in moving forward in pursuing your passion related to research. Lastly, I would like to encourage you to continually visit our website, research.ucdavis.edu, to learn more about how the Office of Research can support you in all your efforts in advancing the research. Following this session, we will provide lunch and hope you tour the exhibits that are set to learn more about the support services and learn about various research groups across the campus. Then at 2:30, we will have several breakout sessions for you to choose from, followed by an afternoon focus on ag innovation in partnership with Seed Central. Now, it's my distinct pleasure to introduce Professor Kimberly Mcillister to moderate our keynote session on enhancing brain health and neural performance. Kim Mcillister is the director of the Center for Neuroscience and a professor of neurology in the School of Medicine and the Department of Neurobiology, Physiology, and Behavior in the College of Biological Sciences. Professor Mcillister has been at UC Davis for over 22 years. You beat me by 1 year. And has tremendous success running the research labs. She has been leading an interdisciplinary team of researchers at UC Davis to study the effects of maternal infection in increasing risk for neurodevelopment disorders, psychiatric illness, and Alzheimer's disease. Their team has brought in more than $25 million in grants from National Institute of Mental Health to establish the UC Davis Silvio O. Conti Center to identify at-risk pregnancies and new treatment to prevent the detrimental effects of maternal infection and brain development in offsprings. Professor Mcillister is the recipient of many awards, including the Pew and Mark Scholars Award, a Sloan Fellowship, and Society for Neuroscience Young Investigator Award. Over to you, Kim.
Thank you so much for that introduction. Is the sound okay? Oh, good. So we are really grateful for the opportunity to showcase neuroscience research here at UC Davis today. This is an auspicious time to highlight innovations in neuroscience, since it is the 30th anniversary of the Center for Neuroscience. The center was one of the first neuroscience centers in the country, and it was the first interdisciplinary center here at UC Davis. It was established in 1992, so about 30 years ago, and since then, neuroscience has grown steadily to become one of the most fundamental strengths here at UC Davis, with more than 300 faculty that are located in 12 centers and 41 departments across 5 colleges and schools across all of our campuses. So this year, we're also celebrating the establishment of the UC Davis Neuroscience Consortium, which brings together these faculty to enhance collaboration and interdisciplinary research. So I would really like to encourage you to check out the breadth and depth of neuroscience. You're just going to get a taste for that today, but there's a lot within this field at UC Davis. So please check out our ucdnc.ucdavis.edu website. So today we are going to focus on 3 of the most exciting areas of neuroscience research here at UC Davis. And we're going to start with a topic that many of us are interested in, which is optimizing brain health throughout the lifespan. This topic is led today by Dr. Charlie DeCarli, who is the Distinguished Professor of Neurology at UC Davis and the recipient of the Victor and Genevieve Orsi Chair in Alzheimer's Research. He is the director of the UC Davis Alzheimer's Disease Center, which is a United States National Institutes of Health funded Alzheimer's Research Center. Dr. DeCarli is also the director of the Imaging of Dementia and Aging, or the IDEA Laboratory. His research focuses on using advanced structural and functional brain imaging to study normal aging, mild cognitive impairment, and dementia, and the role of genetics, diversity, cerebrovascular, and Alzheimer's disease on these processes. So Dr. DeCarli, please join me up on stage.
Thank you so much. Oh, there we go. It's a pleasure to be here. Actually, it's the 31st birthday of the Alzheimer's Disease Research Center as well. We've been funded for 31 years and moved from an ADCC to a research center just recently. So this is a great place to be, you all.
So, Dr. DeCarli, I want to start by bringing up an issue that many of us are concerned about, which is memory loss as we get older. Everyone has heard of and fears Alzheimer's disease, but there are many different types of dementia. So could you tell us a little bit about how age-related memory loss and Alzheimer's disease and other dementias differ from one another and what characterizes them?
Do we have a few hours or a seminar series?
How about briefly?
So, yes, the most important point is to understand that there are different types of dementias. And to begin with, too, is to understand that as we get older, 2 things happen to us. One is we take on more responsibilities. And 2, the most common cognitive differences that occur as we get older is that we were born with a CPU that had only, 16-bit access and not a lot of RAM there going. And so that diminishes as we get older. And so a little bit of forgetfulness, particularly for names of people, or my favorite one is, that Beatles song, the one with, oh, golly, I can't tell you, but it'll come to me, is very, very common. So first off, rest that. But almost all diseases that affect the brain, short of vascular disease, have something to do with abnormal polymerization of proteins. So whether they be the Alzheimer's proteins, which is the tau and the amyloid, the Lewy body proteins, which is alpha-synuclein, TDP43, all of these proteins have a consistency in that they are what cause the neurodegeneration. And they manifest in slightly different ways clinically. And so the classic Alzheimer's, where you have memory, followed by a little bit of trouble with attention and complex tasks, and then a little trouble with language and coming up with the words. What I call empty speech is not just you can't remember the Beatles' White Album, but you can't even articulate that it's the Beatles' White Album, okay? That's Alzheimer's disease. Everything else kind of varies off of that. And it's a bit complicated to say, but I think the key features is understanding these biological processes that lead to the neurodegeneration.
So why do some people get Alzheimer's disease at young ages and other people live to 100 without any age-related memory loss?
Yeah, that's actually right out of my grant for the center. That's what we're trying to understand. I think that the way I think about it is that there are areas of brain development. So in that, Kim and I share this understanding of how well does your brain develop, so reaches its maximum ability, and then how well can it sustain itself to either prevent injury processes, such as Alzheimer's, Lewy bodies, etc., or withstand them. So one's a kind of like a resilience and one is kind of like a reserve where you can take a few hits and still manage to do okay even though the pathology is there. What we believe is happening is that it's the combination of how you developed and then life experience that accumulate over time as, you know, sort of dementia by 1,000 paper cuts. So the more diseases that you have, the more likely you are to be demented. And the notion that Alzheimer's disease or Lewy body disease occurs as a singular disorder is now thought to be passé. Alzheimer's disease is the most common pathology in the brain, but the people who are demented often have multiple pathologies in the brain. So it's Alzheimer's plus vascular disease, Alzheimer's plus Lewy body disease, etc. Younger people who get it before the ages, say, 50s or 60s, those people have specific susceptibility features, either a primary genetic, where I call a mutation, which is definite and causes the disease, from those who have high-risk genetic influences like the apolipoprotein E, for example. So I know that's a long-winded way of saying that, but I think how we get through life begins where we start and how well. So people like Dr. Mcillister are trying to understand how to make the best brains possible, and then how many hits we get and what we do with that.
So we're showing a slide above you that shows the different trajectories of brain health throughout the lifespan. And clearly, as we're talking about, some individuals experience at some point their trajectory of brain health veering away from healthy brains. And so we typically think of Alzheimer's disease and memory loss as things that we need to worry about when we are older. But how can thinking about this healthy trajectory help us to understand Alzheimer's disease and maybe treat it?
Thank you for that question. So the important point is this idea of what's happening in your early to middle life and what can you do to modify that. And in a sense, it fits very nicely in what we understand about cancer, heart disease, etc., is looking at the things that you can do in your life to modify the impact of normal illnesses. And let me take a common medical illness, hypertension, okay? In this group of individuals, just looking across, there's probably between 25 and 40% of you who are either have diagnosed or undiagnosed hypertension, okay? Some of you are being treated very effectively, others are not being quite as well treated. And that kind of thing is what we need to focus on, is reducing those cumulative risk factors that we can control. And that includes lifestyle choices. The big one is, even though people talk about smoking as hazardous to your health, it's really, its role in Alzheimer's disease is not certain. So I just sort of put that aside. It's not good for your heart, and what's not good for your heart is not good for your brain. But other things you can do, like exercise, and then this whole issue about diet. And diet is really important. in that you make lifestyle changes in your diet and you start young. So we really do understand that there are certain cultural diets that have a substantial impact on whether you're going to develop a late life dementia. And that's this quote Mediterranean. And now it's been kind of evolved into a brain health diet. The other things are exercise. And I would say if you were going to pick one thing to do, which we don't like to do, but the one thing is to exercise. And what do I mean by exercise? I don't mean that you have to run a marathon every week. What I'm talking about is 20 to 30 minutes of walking up 3 to 5 times a week. That is all you need. And in fact, there's really good evidence that even modest exercise is quite helpful. Once you get up to, like a professional sports person, it really doesn't give you any more advantage. And the type of exercise, we tend to think more cardiovascular, but we don't really know yet. So I would say any type. That can include yoga, that can include, strengthening as well. So flexibility, strengthening, and cardiovascular, and some mix to that would probably be the best. So as individuals, what can we do? These are the things that we can do with our lifestyle. And then anytime we have a medical illness, to address that illness and try to get into what we call ideal control, whether it would be your glucose, your blood pressure, or your cholesterol. I hope that answers the question.
It absolutely does. So what I'm hearing is that if we haven't developed these healthy habits for our heart or for our bodily health, that if we want to improve our mental health and our brain health as we age, that we should think about doing those now.
Right.
Yeah.
In fact, just so you understand that when you look at people after they've passed and you look at their brains, above the age of 80 years, almost all of them have some degree of cerebrovascular disease. So it's almost ubiquitous. And again, it's not that that's causing the dementia. What it's doing is shrinking that reserve. So anything else on top of that sets you up to be susceptible to the expression of dementia syndromes. And none of us want to have that.
So wouldn't it be great if we could come up with a way of identifying when people veer off onto an unhealthy trajectory so that we could let them know that it's maybe even more important to do these things?
Right. And this leads into the importance of what we're calling biomarkers, so that we are able to see the pathologies that are occurring in the brain before those symptomatologies are evident. And in fact, we've for Alzheimer's disease, we've reclassified Alzheimer's disease to using biomarkers to the presence of amyloidosis in the brain as a preclinical phenomenon. We don't know exactly what it means yet, but we're using biomarkers, and the goal would be is that for every individual, we have a precision medicine approach using these available biomarkers and things that haven't been developed. And here's where I'm going to talk about the clinical translational, and that is, the things we can do are find those markers that would tell us where we veer off. And they could be through passive monitoring, they could be through active monitoring, they could be through new protein measures in the blood, the urine, less so in the CSF, fancy new imaging techniques. There's a host of genetics, understanding, making polygenetic risk scores to see who may be more susceptible based on whatever. And often these polygenetic risk scores interact, so they make you susceptible, but it's because there's some other life factor that's going on that you might be able to modify. So I hope I answered that question.
Absolutely. So it sounds like you're going to need teams of researchers with lots of different areas of expertise to come together to address some of these issues, especially as you get into precision medicine and wanting to come up with new treatments for individual cases. So I know that the Alzheimer's Research Center cares deeply about team science. So can you tell us a little bit about that and about some of the opportunities to get involved?
Yes, thank you so much. So we do care deeply about team science. And in fact, over the 30 years, we have collaborated with almost every department and I know at least most of the schools in the university. And we do that through what used to be called a pilot projects program, where you were given a fixed amount of money. And now we've changed to what's called a developmental projects program. And this development projects program is designed to work like, if those of you are familiar with NIH, are U grants, where it's a collaborative relationship in which we, as the Alzheimer's Disease Center, have set aside about $100,000 a year to invest in scientists on campus to help them develop a career in Alzheimer's or Alzheimer's disease or related dementias, or any unique and novel. And in fact, of the speakers today, everyone has received a grant from our center. So this is something we're very excited about, and we seek to get this out to you all. We also have an Alzheimer's Disease Symposium. Some of you may have actually been there. We do this every year, where we announce our program, and to reiterate how we're here to help, and we look for novel ideas at every level. And then the last thing I want to say before I get too long-winded is we also emphasize training. So we have actually money to have what's called our research educational component, where we help pay for scholars who may be at any level from pre-doctoral to postdoctoral to junior faculty, to help them with travel or education that may be related to unique things that they need to pursue their career.
So, Dr. DeCarli, your center is incredibly well funded by the National Institutes of Health, but I would imagine that that might not go far enough to be able to give you the resources that you need to do this kind of team science. So can you tell us about some of the things, some of the other ways that you're funding this research?
Yeah, so I think that this is a really good point, is that the NIH, as you all probably know, is fairly restrictive. You write a grant, you have to do these things. It does not leave a lot of creativity. The NIH is a fairly conservative government entity. So what we look for are foundational grants and philanthropy to help us, and where we emphasize. So I'm, for example, the ORSI chair. My chair goes to training grants. And my goal with training grants is to encourage young people, because I'm getting to be an old fart, and I want more people to come in and follow me and become exciting scientists in this area. So where can we be the most effective are when we get these foundation grants or philanthropy, where we can do things to encourage training, but also to do high risk, high reward research. And I think that's something that we try to emphasize. We're doing a little bit of that. We set aside a little bit of that of our developmental award to pick one high risk project, but that's where we can have the biggest impact. You know, I think a lot of times people talk about in philanthropy, I want a building. I don't care about a building. What I want to do is be able to have a little bit of stash to do these interesting things and to train up people. So that's where I ask my, who are thinking about giving to this, to focus their energies.
Great.
Unless if they want to buy me a building, that is absolutely okay.
Right, you have to have space to house all of these amazing researchers
Yes,
together.
that's true.
So, yeah,
That's
yeah,
true.
Yeah. So you talk about high risk, high reward. Can you give us a flavor of what some of the most exciting directions are to you right now if resources were not an issue?
Were resources not an issue?
In Alzheimer's research.
In Alzheimer's disease.
Yeah.
I think that I take a step back, and that is what I would believe would be the most exciting would be understanding the basic mechanisms of neural development, connectivity, and sustaining that connectivity. And it'll be great because we're going to be having a conversation about that. And I think this is high risk, but potential high reward. And I believe that, of course, anything that translates into effective therapy. So we are really positioned to understand the basic mechanisms of Alzheimer's disease in particular, but we fail to translate those into effective treatments, even though you probably have all heard about aducanumab. That is an early kind of brute force. I'm going to just grab these abnormal proteins and pull them out of the brain. I would rather prefer that they not ever get in there in the first place. And why does that happen? Well, we certainly know it has something to do with the membranes of our cells and how those cell membranes change with age. So I think there's a whole area out there that's rich for investigation, but it may go nowhere at first. And so that's where I think we should be focused.
Right. So you're talking about basic science and bringing basic science into developing companies and developing products.
Right. And
Great.
I thought that's why we're here today.
I think that is why we're here.
Okay,
today.
good.
Yeah, great. Okay, so before we let you go, we've got about 5 more minutes. I know that we would all benefit from hearing about what you've learned along your impressive career. Again, we've got about 7 minutes. You've learned a lot. And if you'd frame it to answer the question of what advice you might have for some of the researchers that are in the audience that are just starting out, some of the students here.
Okay, I would say the one thing about my career that I continually reflect upon is finding mentors. So if you're a student, find somebody that you think you want to become and visit with them. And, I guess my time's up. And visit with them and pick their brains. And it's okay to mimic somebody who's doing a good thing. Across my experience, I have a handful of mentors who stepped in at every part of the way to make me successful. I'm sure some of them wished I was a little more successful than I am, but that they were keen. The second thing, and this is the second lesson I've learned, is do something that you're passionate about. I know sometimes researchers get into a place where their current line of research isn't sexy for the NIH, so they try to change gears. That's okay if that changing gears is in something that you understand and wish to grow in that area. Because right now, for example, the NIA went from a $1 billion a year institute to a $3 billion and hopefully will be a $4 billion a year institute. That we hope that encourages all of you to get on board and join us in our fight. However, if it's not your stick, don't do it, okay? And then the last thing I would say is persevere. Man, reviewers suck. I mean, and it's funny because I've been a chair and I've been an editor, and it's so funny, somebody will come up, I know who reviewed that, and I'll say, oh, okay, and they'll tell me about somebody they hate, and actually it was one of their friends. So persevere, because everybody has their own ideas, and I just take Stanley Rappaport in the Laboratory of Neuroscience, where I trained at the NIH, said to me, when we'd get an article rejected, he'd say to me, good. What, Stanley, come on, man. And he'd say, yeah, that means we're smarter than the reviewers. So hang in there. It works. It's hard sometimes. It can be very challenging, but we're doing the good work here.
Well, I could ask more questions, but I think that's a wonderful place to end this conversation on. Thank you so much. It's really
You're
for all
welcome.
the work that you do. Terrific. Okay, so our next topic is switching gears, and now we're going to talk about neuroinnovations, restoring, augmenting, and extending human capacity. And our next speaker is Dr. Karen Moxen. So Dr. Moxen is a professor in the Department of Biomedical Engineering here at UC Davis. She has conducted groundbreaking research in neuroengineering, developing computational models to study the encoding of sensory and motor information. An important focus of her work is the impact of neural injury on the representation of information in the brain. Early in her career, she contributed to the first demonstration of a closed-loop, real-time brain-machine interface system in a rat model that was quickly translated to non-human primates and more recently to humans with neurological disorders. This work has spurred an entirely new discipline within neuroengineering, and it has had and will continue to have a global impact. So, Dr. Moxen, we're very excited to have this conversation with you. Thank you for doing this.
No, great. Thank you for setting this up and that kind introduction. And I feel like COVID is waning and it's springtime, and it's just wonderful to see you and to be able to be here and talk about things. So great. Nice to be here.
Terrific. So I want to start simple, but with something that might be confusing. So you are a neuroengineer.
Yes.
Before we get started, can you tell us what that means?
Sure. I think we all agree that in the entire universe, there is no more fascinating a system than the brain. On the one hand, we know so much, all of the things that Dr. DeCarli just went through, all of the things that neuroscience has brought us in the last, millennium. But at the same time, there's so much that we don't know. And why wouldn't engineers want to get in on that? I mean, it's just incredible. When you get introduced to the brain, you're just like, oh, I didn't know about that. That's what I want to be a part of. So if you think about the whole spectrum, labels are useful, and the label neuroengineering is useful, but I'm very broad about the topic, right? It can be, if you feel like you are a neuroengineer, then you're a neuroengineer. So, but think about the spectrum of neuroscience and neuroscience research. On one end, there's your traditional neuroscientist who might go into the lab and say, I have a hypothesis about how I think things work. I'm going to develop an experiment to test that hypothesis, and I'm going to take those data, and I'm going to interpret those data to try to understand something better about how the brain works. On the other hand, far end of the spectrum in neuroengineering, I'm an engineer. Engineers typically were problem solvers, right? I see a problem, I want to fix it. So spinal cord injury, tragic, tragic problem. How can we use our engineering know-how and solve that problem? So we would take everything that those neuroscientists bring to us, we would take everything that we know about physical systems that we learn from physics and math, and apply that and create a solution. But I would say that neuroengineers exist on that entire spectrum, right? You can use the techniques and technologies that we've developed in neuroengineering and create new kinds of experiments that no one had ever conceived of before because of all of these advances in technology, and all of a sudden, you can study the brain in ways you've never been able to study the brain before. So some people, background and training is neuroscience all the way. They have a biology undergraduate, they have a PhD in neuroscience, and they are clearly neuroengineers. Other people on the other spectrum, they have studied engineering the whole way through. They interact with and collaborate with neuroscientists and other important investigators, and they are neuroengineers. So I think as Dr. Carli said before, what's the advice to young people coming up? If you're passionate about it, go do it and embrace the label if it helps you, and don't worry about the label if it doesn't help you. So that's kind of how I feel about it.
Terrific. So in 2017, you published a paper highlighting a novel approach that allowed paralyzed rats to walk again, which is amazing. So can you explain the approach that you developed and what the implications are for this kind of intervention in humans?
Yeah, so again, I'm back to, the brain is just the most fascinating system in the world. And I was very attracted to that from the very beginning. And early in my career, I worked together with people who were very passionate about understanding spinal cord injury. And you could imagine that if you're going to study spinal cord injury, where are you going to put all of your effort and all of your focus? You're going to put it on the spinal cord. I want to repair the spinal cord. And that's important, and there's a lot more work that needs to be done there. But there's two other very important things to think about. One is that a spinal cord injury is also a brain injury to some extent. the neurons that live in your brain send axons all the way down your spinal cord, and then they synapse into the network in the spinal cord that sends signals out to your muscles on how you want to move. If you have a spinal cord injury, those axons are damaged, and they impact those cells that are living in your brain. So we can't forget about the fact that a spinal cord injury is going to have that kind of an impact on the brain. And then from another point of view, we all now appreciate clearly that the brain is plastic, right? There was an idea that, well, you go through a young age and we have all this plasticity and we learn of all of our things, then sometime after adolescence, it all goes away and we don't learn anything new and we just have this plateau for the rest of our lives. And we know for sure that that's not true anymore. The brain is extremely plastic. So if you have an injury like a spinal cord injury, your brain says, hey, I still want to move. I still want to get up and do things. How can I change? How can I have plasticity in my brain that's going to optimize my output? So that was sort of the insight into what we tried to do. And then we said, okay, how do we develop therapies that will engage the brain and induce that plasticity? So we did it with a combination of therapies. So two things sort of came out of this. One was that spinal cord injury, in order to optimize outcome, you have to consider the neural system along the entire neural axis. And that means from the brain all the way down to the nerves that go out to your muscles. And the second is that if you want to optimize your output, you have to bring together many technologies. So we brought together pharmacology and rehabilitation. And by combining all those together, we were able to get animals that had a complete spinal transection to get up and take weight-supported steps, which was the first time that had been demonstrated in that kind of a model. But I think more importantly, what we found was that now rats are different than people. So these experiments were done in rodents. And if you have a healthy rodent and you go into his motor cortex and you lesion the motor cortex, they can still get up and they can still walk. They're perfectly fine. They have some minor motor deficits, but they don't really need their cortex to walk. It's not true for humans, but for rats this is true. If I take an animal that has a complete spinal transection and I provide therapy that allows the animal to get up and walk, and I go into that animal's motor cortex and I lesion that motor cortex, all of the gains and benefits that we have made are gone. So we know that post-spinal cord injury, those changes that we're seeing in the brain, especially those changes that are induced by therapy, are critically important for that functional outcome. And I think that's made a big impact in our understanding about what we need to do to really optimize performance in spinal cord injury.
It's incredible. So some of the work that you do really does seem straight out of a science fiction movie because it's so cutting edge and forward-looking. And I know another area that you focus on in your lab is the brain-machine interface. So an acronym for this is BMI. We're not talking about the BMI that you hear about in the doctor's office, but the brain-machine interface. So can you explain what a BMI is and how it works and why it's important?
Sure. So if you think about what I was just saying in the sense of no single one therapy is probably going to improve function after spinal cord injury. We need to bring together multiple therapies. So we spent a lot of time with pharmacological therapies and combining them with physical rehabilitation, and we really only got so far. And being an engineer, we said, okay, what can engineering bring to the table? So if I can improve performance to a certain level, but my inability of my brain to communicate with regions below the level of the lesion, then maybe I could, like a little telephone line, jump around that lesion and take signals from the brain and have them control systems below the level of the injury. So brain-machine interface is this idea of putting electrodes into the brain and recording the activity of neurons and decoding what the intention is. So for many years, many of you know about EEG, right? Electroencephalography. You can put electrodes on the surface of your scalp and you can record those signals and you can understand several things about what's going on in the brain and see all kinds of different signals. But those signals are really far away from the source of the activity and the information content is strongly degraded. So brain-machine interface comes along and says, to really get the information out of the brain, we have to stick electrodes into the brain. So we took large arrays of microelectrodes. What's important to understand here is that in order to be able to do that, we had to have huge technological advances. So let's say in the 60s, people could put in one electrode at a time and record from one neuron at a time. And you could say, okay, if I put that neuron into the sensory cortex for the hand and I tap you on your hand, then I can see that the neurons are changing their firing rate. So somehow that firing patterns of neurons is encoding the information about where you're contacted. If I put electrodes into the motor cortex and I ask you to move your arm, the pattern of a single neuron is going to change its firing rate depending upon how you move your arm. So we get this general idea that single neuron firing patterns are conveying information. And in around the 70s, people said, okay, I can't really record from many neurons at a single time, but I'll have the subject repeat the activity over and over again. And then I will just say, well, what happens if I pretended that I recorded a bunch of different neurons across all those different trials? And it looks like I can probably tell you what direction the subject moved their hand. So we knew that there was enough information in there on a single trial to tell you what you wanted to do. Now technology evolves. We get PCs on your desktop, and all of a sudden people now take stacks of personal computers, each one controlling a single microelectrode that's put into the brain, and now we can simultaneously record a whole bunch of neurons at once. And the reality is that in those patterns of neurons across many cells, we could, on a single trial, tell you what direction you moved your arm or what type of stimulus that you received. And that really kicked off, I think, what is the beginning of what we think of as brain-machine interfaces now. And in the 90s, they began to build dedicated systems for this. And I think that technology advancement was really critical. So now you have these single boards that can each record, let's say, 8 neurons at a time with all the filters and amplifiers, because you have to realize that those little neurons are producing microvolt-sized signals. And they are next to thousands of other neurons that are also producing microvolt-sized signals. So we need to amplify that and filter and actually know when a single neuron is firing an extra potential, and we need to do that for hundreds of neurons. And once you can get to that point, now it's becoming actually relatively easy. The information is there. We do a lot of machine learning algorithms and other types of computer science approaches to improve those decoders. And now we have humans controlling robotic arms to pick up objects, pour a glass of water, bring it to them, and drink. These are people who were paralyzed and not able to move for 10, 20 years. And we implant these little tiny pin-sized devices in their brain that record hundreds of neurons, take that signal out, decode it, use it to control the robot arm, and people can now feed themselves for the first time, give themselves a drink of water for the first time. And if you go to YouTube, there's a lot of videos out there showing patients who have been implanted and what it's like for them to use these types of systems. And you can see the joy in their eyes that they can finally start to take care of themselves a little better than they could before. It's quite moving.
Oh, it's remarkable. Absolutely remarkable. So given how quickly technology has been moving, where do you see this field going in 5, 10, 15 years? And how do you see that impacting clinical care?
Yeah, so that's a really great question, and it is complicated. If you think about the neurotechnology industry today, excuse me, it's projected to be about a $15 billion industry in 2024. Twitter has a market cap of 36 billion. So what are we talking about here, right? And a lot of that has to do with, what does it mean to translate science and technological advances that you can do in the research lab to patients in the clinic. And it is just tremendously harder than you could imagine. And part of it is the resources, right? So where do we get the resources? If I show a demonstration, because the NIH and several other foundations and private philanthropy provided enough money to develop these technologies, and now there's maybe 20 patients implanted with these devices. It's several $1 million per patient to get this going. Now there's a long trajectory of how to make this affordable, how to improve the technology so that we can have high reliability and low, potential for something to go wrong. This is a big, they call it the valley of death, right? We do these great things in the lab and we say, boom, why shouldn't people be doing this? And if you look back through the history of neurotechnologies, it's kind of a sad story. We have developed devices that can stimulate your muscles and restore function in people's hands who have lost it due to some kind of injury. And people went off and spun off companies, and those companies got started, and they built devices, and they provided them to the community, and there was no way for the people to pay for those, and then for that enough money to come in to keep advancing those, and those technologies, those companies fell by the wayside. What's the likelihood that these technologies will really start to take off and people will recognize the cost benefit, right? So if I'm a paralyzed person and I can't move, I'm a tremendous cost on society. My brain is working pretty well if I have spinal cord injury, but I'm not able to contribute to society because I'm locked in at home and I need 4 or 5 caretakers. So we know the cost benefit analysis is there. That's crystal clear. But it's always that first hurdle to get over to get the technologies cheap enough, not so expensive so that we can have more and more people using it, and then that just creates a sort of a snowball effect where we can then create more technologies. So if I had a crystal ball, I would love to say that this is just going to continue to accelerate like you see accelerations in other forms of technology, right? They're not a linear increase, they're an exponential increase. And I think neuroengineering is starting to hit its paces where we will start to see that kind of exponential increase in the years to come.
I hope so. I really hope so. It's so exciting. So are there any concerns about the misuse of BMIs or unintended consequences?
Sure. I mean, if you think about, so who wants to get into this space, right? I don't know, many of you may know, Elon Musk's got a company called Neuralink, where he would like to implant electrodes into your brain and have you plug into the computer every day, even if you're perfectly healthy. Nissan and a couple of other car companies have programs to record your EEG signals because they're concerned about you falling asleep behind the wheel of the car. And their point is, if you fell asleep behind the wheel of the car, I could record your EEG signals and alert you if you're about to fall asleep. But what are the unintended consequences of this sort of thing? And if you just think about Facebook for a second, what happened when you could just click on a like symbol, a thumbs up or a thumbs down? The massive amount of information that they could get about you personally and use to, I would call it manipulate you with advertising to make you buy things you don't want or you don't need. It's amazing, just those little clicks. Now consider the fact that your neural signals are being recorded 24/7 or maybe 8 hours a day. This data is going to some database somewhere, and now they're really profiling you. So I think that's one of the greatest concerns is what happens to all this data and what are we talking about with privacy? What does it mean to own your own data and who gets access to it? And if I want to, it seems as though advertising is the thing that drives everything moving forward. If we're going to rely on advertising to move forward the neurotechnology space, we could be in trouble because who's getting control of the data then, right? So I think those are all things that we need to think about and be concerned about. And then there's another side, which is agency. If I am using my neural signals through some sort of decoder, that decoder is not passive, right? It's passive, but how you design that decoder can change what the outcome might be for a certain set of neural signals that you put into it, right? So you have to have a sense of agency. We all consider ourselves, when I want to move and pick something up, I am doing that. Now you're taking your neural signals out and you're moving a robotic arm. What if that robotic arm is not doing exactly what you intended, but it's okay? How much agency have you lost, and how much does that agency influence you as a person? We have examples from Parkinson's disease, for example. You may or may not know, you sort of start out with a drug regime with Parkinson's disease, but eventually most people will have a Parkinson's stimulator chronically implanted into their brain, and it will constantly stimulate. And there are plenty of examples where that Parkinson's stimulator has changed people's personality. Why this happens, we don't exactly know. But then what does this mean in terms of agency, right? So if it tends to make you much more gregarious, so outgoing that now you become a risk seeker, and you take your family fortune and you spend it all, and your family comes in and says, turn off this stimulator, it's changing our loved one, and they are not behaving in a way that we like, and that person says, I feel great, I don't want you turning off my stimulator. What is the right thing here? These are very complicated, deep questions that need a lot of thought, and I'm sorry, I don't have any answers to them, but the level of misuse is huge, so I think we need to have really long, deep, strong conversations about that.
So it sounds like to be able to move this forward in an ethical way, that you need ethicists on these teams, as well as neuroscientists and engineers. You also need industry involved. You need a lot of different players. And I know that you've been visionary in trying to bring together all of these different types of faculty members and trainees in a new center that you've established here, the Center for Neuroengineering in Medicine. So can you tell us a little bit about that and what your vision is for the center?
Yeah, so this is very exciting. So Kim, in the beginning, you put up the Neuroscience Consortium. So if you go to that Neuroscience Consortium site and you look at the centers that's a portal to open to, one is the Center for Neuroengineering in Medicine. And I have to give, a shout out to Professor Sanjay Joshi in the Department of Mechanical and Aerospace Engineering, who was the original sort of driving force behind it, and his efforts actually helped recruit me here, and I will be forever in his debt for that. And it took a few years after I got here and a few other people, we sort of created a triumvirate. So Sanjay, myself, and Carolyn Patent, because it's a lot of work, and neuroengineering is extremely broad. So Carolyn Patent is a professor in the School of Medicine, and she's very interested in translational research, especially as it relates to improving clinical practice. And together, we created the center, we got support from the Office of Research, thank you very much for that, and the College of Engineering, which we couldn't have functioned without it. And we were able to bring in Gabriella Lee, who's sitting here in the audience today, who has experience running centers at UC Davis in the past, and she's our associate director. And together, we've been able to launch the center, which was very important because it gave us space for everybody to come together. And you're exactly correct in this idea that it's not just these people called neuroengineers. Some of us are core sort of neuroengineers, but we do have ethicists, we do have regulatory people, we do have people in the legal department, we have people who are basic fundamental neuroscientists who are interested in collaborating with us. We have people who are mechanical engineers, people who are electrical engineers, and they would definitely consider themselves electrical engineers, not neuroengineers, and they contributed. So it's a highly interdisciplinary program. We have members from all of the colleges on campus, and work, collaboratively with, of course, the National Primate Research Center here at UC Davis, the Vet School, School of Medicine, as I said before, and being able to bring all these diverse people together and for them to have a place where ideas can be exchanged. And I think that's the most important thing, and a place where you can go and look and say, I need a collaborator on this project. Where can I go to get that collaborator? And one of the things that's exciting is, so we have 4 research thrust areas. One is sort of neurotechnology and neurocomputation. One is human-machine interfaces, and you could imagine these are very important in high-risk environments. So astronauts who go out in space, or firefighters, how do you develop machines so that when you're in these high-risk environments, those machines are working closely and interacting well with the user? And then, of course, we have neurorehabilitation. And the final one is what is new. It's brand new concept. And UC Davis is the first sort of to put it forward, and it's something I'm very passionate about, is cognitive neuroengineering. So, Dr. DeCarli has been speaking about Alzheimer's disease, but there's many other neuropsychiatric disorders that could benefit from neurotechnologies. So, for example, depression scales, we know that if you do some neuromodulation, you can improve outcomes from depression. Well, what if you could record your neural state, and as you begin to move to a depressive state, we could do a little bit of neuromodulation and just push you out of that state and push you back into a more, normal range of states. Or vice versa, you're a high-risk gambler, and we want to, you want to get out of that, and drugs have side effects, and sometimes drugs aren't always the right way to go. So you implant a stimulating device, and when you see yourself, or when the system picks up that you're moving into a space that is inappropriate or beyond what you would like to be, you could sort of stimulate and come back to a more healthy range of cognitive function. So cognitive neuroengineering, go to our website and check it out. It's a new field, and UC Davis is really, as you pointed out in your introduction, we had the first Center for Neuroscience. The cognitive neuroscience on this campus is bar none the best in the country, and taking them and combining them with our engineers, it's been a really exciting journey. And we're planning on trying to go to the National Science Foundation and get a Center for Neurotechnology and Science to support this cognitive neuroengineering endeavor.
Awesome. It's very, very exciting. So we have about 3 more minutes, and I want to bring up a topic that I know is near and dear to your heart. So one of the things that strikes me in these centers, and especially in your center, is that you need as much diversity of thought and ambition as possible. Mm-hmm. And so I know that in STEM and in engineering in particular, there hasn't been as much diversity as we'd like to see, and as you might need to really make one of these centers successful. You've been a major proponent for supporting women in engineering, so can you tell us about why that's important to you, and maybe leave us with some words for some of the trainees in the audience about why this is exciting, and you should never have a barrier to getting into a field?
Yeah, so this has been hard in engineering, really hard in engineering. And so we put together an organization we call WINE, Women in Neural Engineering. And it's been for twofold. One is we have a pull, we have a need. And I'm constantly just saddened by seeing these young women come through and working hard to get their degrees and graduate. And then it's not any one thing, it's this death by 1,000 cuts. Little things that happen, we tend to be more family caregivers, we tend to be the one who take the biggest burden for, having and raising children. And when you add up sort of all those little things, it just becomes harder and harder to stay in the neuroengineering field. And I think part of that is because we have this sort of, especially in engineering, this sort of little round circle that says, if you can fit into this little round circle, then you can be a member of neuroengineering. And the reality is this is bad for neuroengineering. It's bad for engineering in general, and it absolutely makes no sense. How could we begin to talk about developing new technologies to help people with neurological injuries and disease when half the people are women and 99% of the people studying this are men? It makes no sense. And then when I'm also continually surprised when I talk to women, and there is this very sad fear of being considered a token. And I'm just want to say to everybody, your contribution is valuable. And I think Dr. Carly said this earlier, go for what you are passionate about. And none of that other stuff matters. And if it has been okay for the last several hundred years for a small segment of society to be in control because it was passed down from generation to generation, that to me is the greatest sense of tokenism I've ever seen. So how can those people who have been in control all this time say, oh, well, somehow I'm not a token and somehow you are a token? It's absurd. So I say to women out there, there's no such thing as tokenism. It does not exist, or it has always existed. And that's it's either one of those two extremes, and there's no in between. So it doesn't matter. You belong here, you're wanted, and you're desperately needed, and you have a contribution to make, and focus on that, and all the rest of it is not important. When you need support, look out and get it. I think this is very important what Dr. DeCarli said. Find a mentor. It's the most important thing. Some people who have power in neuroengineering don't necessarily make the best mentors, so you want to find someone who you think would make a good mentor for you, and you probably need more than one. No one person is probably going to be able to understand all of your dimensions, And each one of those dimensions of who you are, whether it's culturally or gender or just passion about different aspects of science, you'll probably need a bunch of different people. And I think it's smart to say, okay, this is who I am, and I am this multifaceted person, so I'm going to find 3 or 4 mentors and use them in different ways to give me insight into the pieces that I need to create the kind of career that I really want to have.
Terrific. So I know that within the new Center for Neuroengineering and Medicine that Dr. Moxen has lots of really innovative ideas for how to enhance diversity and support all sorts of different kinds of perspectives in this field. And she's really also a dedicated mentor, as is Dr. DeCarli and our next set of speakers. So thank you so much, Dr. Moxen, for everything that you do.
It's a great pleasure. Thank you.
Okay, so finally, our last topic focuses on an equally interesting topic. It's on using psychoplastogens to selectively rewire pathological neural circuits. And this area of research is being pioneered by Dr. David Olson and by his collaborator, Mark Russ. So I'll introduce both of them now. So come on up to the stage. Thank you. David Olson is an associate professor in the Department of Chemistry and the Department of Biochemistry and Molecular Medicine. Professor Olson's expertise spans central nervous system medicinal chemistry, molecular and cellular neurobiology, and behavioral pharmacology. His academic lab discovered that psychedelics promote structural and functional neuroplasticity in the cerebral cortex. They coined the term psychoplastogen to describe small molecules that produce rapid and long-lasting psychedelic and ketamine-like effects on neuronal structure. And they invented the first non-hallucinogenic psychoplastogens that are capable of producing sustained therapeutic effects in preclinical models after a single dose. In 2019, Dr. Olson co-founded Developmental Therapeutics, a company developing non-hallucinogenic psychoplastogens for treating brain disorders such as depression, post-traumatic stress disorder, addiction, and Alzheimer's disease. He is an associate editor at ACS Chemical Neuroscience, and he's received several awards, including the Geordie Folk Pye Award from the American Society for Neurochemistry, the Sigma Xi Young Investigator Award, the Life Young Investigator Award, and the UC Davis Chancellor's Innovator of the Year Award in 2021, among many others. We're also delighted to have Mark Russ joining our conversation. Mark Russ is the Chief Executive Officer for Developmental Therapeutics, located in Massachusetts. Developmental is pioneering efforts to understand how reversing cortical atrophy and rewiring pathological neural circuits can drive rapid and sustained healing for people that are suffering from many psychiatric and neurologic disorders. Mark is a globally experienced life science executive who most recently served as an entrepreneur in residence and advisor at Atlas Venture. Prior to Atlas, Mark served as Group Vice President of Neuroscience at Shire Pharmaceuticals. And over the course of 13 years, he led teams across the United States, Europe, Canada, and Japan at multiple clinical and commercial stages. He was nominated by Pharmaceutical Executive Magazine in 2017 as one of the top 11 emerging leaders in the life sciences. So we are very excited to have both of you join us for this last part of our panel today. Okay, so let's start with neuroplasticity. We've heard a lot about this topic. So many of us, as you've heard, grew up thinking that our brains are hardwired. So as we grow up, they're plastic, and then you hit some stage during adulthood when you can no longer rewire your brain. But as we've heard about today, we are beginning to understand in neuroscience that this is not as true as we thought it was before. So your research program really taps into targeting plasticity to selectively rewire circuits that are damaged in brain disorders. So David, can you start by describing the contribution of neural circuit dysfunction to disease and what brain disorders this might relate to?
Sure. So it's important to remember that activity in specific circuits controls behavior. And in many brain disorders, there's a physical change in the brain, particularly in the so-called stress-related neuropsychiatric diseases. These are things like depression, post-traumatic stress disorder, and substance use disorder. And there's a part of the brain that sits right about here. It's called the prefrontal cortex. It projects to a whole bunch of other brain regions that control mood, motivation, fear, reward, cognition. And so you can imagine if there's dysfunction in the prefrontal cortex, you can have a range, it can cause a range of disorders. And in many of these cases, including the neuropsychiatric diseases, but also neurodegenerative diseases like Alzheimer's disease and others, the prefrontal cortex physically atrophies and kind of shrivels up. And so what we were really trying to do is to use a pharmacological agent to rapidly regrow these neurons so that they can communicate more effectively with other parts of the brain and alleviate the symptoms of these diseases.
Great. So can you define neuroplasticity for us and how it works?
So neuroplasticity is a very broad term, and if you ask 10 neuroscientists what it means, you get 10 different answers. And so the kind of neuroplasticity that I'm really referring to here is typically called structural neuroplasticity, the physical changes in the structure of neurons. And those changes in structural plasticity can often lead to functional changes, how those neurons actually communicate with other partners. And so when we're talking about the effects of a psychoplastogen, we're really talking about the physical rewiring of the brain.
Okay, terrific. So David, you and other labs have revealed that psychedelics and related compounds are exceptionally good at promoting structural and functional neuroplasticity in cortical neurons. So can you tell us about how this discovery came about?
Well, it was a long time coming, I'll say that.
Yeah.
I'd say the beginnings of this came when I was a postdoc at the Broad Institute, the Stanley Center for Psychiatric Research at the Broad Institute at MIT. And this was a time in neuropsychiatry where there had been kind of a dearth of innovation. And everyone was getting really excited about this molecule called ketamine. And ketamine is an old molecule. It was been used by people as a dissociative anesthetic for many, many years. It's also known as Special K. It's a drug of abuse. Mm-hmm. But psychiatrists found that ketamine could really, really rapidly produce antidepressant effects. And now today, ketamine is an FDA-approved treatment for depression. At the time, when I was a postdoc, it wasn't FDA approved, but we were all very excited about the mechanism of ketamine. And it seemed that ketamine had this effect on cortical neurons to rapidly regrow these neurons. Now, something about ketamine that really stood out to me was the fact that it seemed to work across indications. It worked for depression. It also worked for PTSD. There was some indications that it could work for substance use disorder. And so ketamine is far from a perfect drug. And so when I started my independent career at UC Davis, I was really looking for molecules that could be better than ketamine, but produce ketamine-like effects on neuronal structure. And that's what really led me to the psychedelics. There was really rich literature, dating back to the 70s, clinical data suggesting that molecules like psilocybin and LSD might work for a lot of the same disorders that ketamine works for. Alcohol use disorder, depression, PTSD. And so the first question that we wanted to address was, do psychedelics produce ketamine-like effects on cortical neurons? And if so, could they offer some advantages over ketamine in terms of their circuit-level selectivity? Could they be more specific and avoid some of the other issues associated with ketamine?
Okay, so you're talking about the use of psychedelics for potential therapies. So Mark, in thinking about some of the ways that drugs are developed and used, what are some of the downsides of using traditional psychedelics in this respect?
Yeah, thank you. Great question, Dr. Mcillister. And before I start, can I just say one thing? This is the first time I've been to UC Davis. I went for a run this morning along that arboretum. You guys have a beautiful campus here. This is a really, really nice place. You're very fortunate to have it, and walked around last night. So kudos and thank you for having me. The promise of early stage research and the promise of what's discovered, I think it was Dr. Moxen who so aptly said, you cannot underestimate or overstate the challenge with which actually bringing a therapeutic, a neurotechnology, or any other type of invention to human use, especially in neuroscience, can be. And psychedelics are no different. So the promise that was so prevalent, and particularly since the early 2000s in the research David referred to with ketamine that eventually resulted in an approved medication, Spravato, opened the doors, but also opened the doors to many more questions about the ultimate scalability. And the simplest way I'll describe it from a scalability perspective is think of scalability from the earliest of stages. So that's things like IP. Many psychedelics that offered promise are either naturally derived products or ones for which there is no possible or potential IP. And there's different schools of thoughts on whether that's a good or bad thing, but it's nevertheless a complex thing to navigate. Manufacturing, API, active pharmaceutical ingredients. These are very difficult challenges to actually physically make, either plant-derived, ethnobotanicals, all the way through to synthetic psychedelics. And there was, while there was promise, not a lot of clear answers on that. A host of other early stage challenges I could list, and then think about a second category at the latest stages, commercial stages, human stages. The way I tend to think about these things in the pharma space or the biotech space are called the 5 Ps. How can you solve for the 5 Ps? First and most importantly, patience, right? Is all this work, is all this money, is all this time and effort actually going to lead to a meaningful patient benefit? That's clearly the most important thing. Secondly, payers. Who's going to pay for whatever the innovation is? Again, that's something we've heard other speakers refer to and cannot be underestimated. Third, physicians, prescribers, or any other practitioners if it's not a prescribing-based treatment. How convenient, how complex, how accessible, how much time is required? In the psychedelic space, many of these promising treatments require inpatient treatments. So many of you may have read about or heard of the promise of psilocybin, magic mushrooms, or LSD, or ketamine, as David referred to. 8 hours in inpatient, 5 hours, 4 hours, time to onboard, time to offboard. These aren't bad things. Lots of good patient outcomes can come from that effort, but scaling it, very, very difficult. Fourth, pharmacists. So dispensation. How do you actually get, in this case, a therapeutic to a patient? And at-home delivery, at-home access, medicine cabinet is something that is very promising and obviously makes a difference in terms of adherence, something that first-generation psychedelics, at this stage at least, cannot do. And then fifth and finally, the 5th P that I think about, policymakers. How accessible can a potential new treatment be in the policy realm, and what can it offer to enhance other policy aims? So first generation psychedelics, the likes of which ketamine is one example, but think of all the others, clearly an exciting space. Clearly we're rooting them on because we think they can and will help many patients if and when proved and approved by the FDA and around the world for therapy. But scalability, major question. And that's where Delics began to come in.
Well, and so that led to addressing some of these issues that you bring up for the traditional psychedelics has led to a second and third generation approach. So could you tell us a little bit about those?
Sure. The simplest way to describe it is first generation would be anything kind of plant-derived, natural, okay, everyone put up your hand if you've, no, just kidding. So the first generation psychedelics were really the ones that formed the foundation for this movement. A lot of mission-oriented researchers, investors, and others who a lot of us are standing on the shoulders of after 20, 30 years of hard work to progress the research, progress in the destigmatization efforts, et cetera, but essentially unmodified from their natural state. The second generation are therapeutics that companies have formed around to attempt to solve for some type of IP differentiation, as well as their PK/PD profiles, so their pharmacodynamic, pharmacokinetic profiles. Can you find a way to perhaps lower what is traditionally an 8-hour experience to a 4-hour experience, or a 3-hour, or a 6-hour? In some cases, how can you lengthen them to make them more intense for those for whom the dissociative, mystical, or spiritual experience are deemed to be part of the therapeutic benefit. How can you formulate them in a different way to be ingested or cross the blood-brain barrier in a more effective way? So these are tweaks to natural first generation psychedelics, but they're steps, but perhaps not leaps. Third, and what we like to believe are final generation psychoplastogens offer a completely different approach, one that decouples the dissociative, hallucinatory, and other elements as well. We're often known as the company that's taking the fun out of the fungi, but there's actually a lot of other very critical, important elements that you need to think about. 5-HT2B agonism, for example. There are on some chemical scaffolds particularly serious risks, cardiac risks. The ibogaine scaffold, for example, is one in which people have died, taking cardiac treatment. Taking ibogaine treatments in Mexico, abroad, for various serious, very important reasons. Military veterans, many of whom I've spoken to, gained a lot of benefit, but you need an actual cardiologist with you, during these treatments to ensure it's safe. We're not only removing the dissociative elements, we hope to improve the tolerability for a lot of these second generation psychedelics as well.
Awesome. So David, I have a question about how your approach and your goal in developing psychoplastogens to treat psychiatric disorders, how does that compare to the treatments that are used right now that are focused on altering the levels or the function of individual neurotransmitters?
Yeah, so the most common medicines used for depression are things like selective serotonin reuptake inhibitors. Probably the most famous is something like Prozac. Mm-hmm. And the way that these molecules were believed to work for many, many years is that they block the reuptake of these key neurotransmitters like serotonin, and that results in very high levels of serotonin in the synaptic cleft. And people thought that rectifying this chemical imbalance might do something for depression. But that really kind of led to the so-called monoamine hypothesis of depression. But there was an issue with it. The issue is that things like SSRIs drastically increase serotonin levels immediately, but it takes weeks to months before patients experience any kind of benefit. And so after years and years of really hard research from people across the country and across the world, what we've now realized is that things like SSRIs seem to promote neuroplasticity. They're just pretty bad at it. They do it slowly over time. And so if you track that neuronal growth phenotype that I talked about in the cortex over weeks, the course of weeks to months with an SSRI, you'll see that patients get better at about the same time that their neurons regrow, not the same time that their serotonin levels go up. And so that again has led to the so-called neurotrophin hypothesis of depression, which again comes back to this idea of neuroplasticity. Now, the difference between something like an SSRI and a psychoplastogen is kind of the speed at which these work. SSRIs promote neuroplasticity through kind of an indirect mechanism that takes a long time. Psychoplastogens do this more directly, and within 24 hours can regrow some of these neurons to produce long-lasting changes in behavior. And for me, this really represents a paradigm shift in the way we think about treating these types of disorders. Previously, patients would be taking things like SSRIs every single day. And daily administration of any kind of therapeutic is going to come with a host of side effects. For SSRIs, it's things like weight gain, sexual dysfunction, etc. And that can, really decrease the tolerability of those types of medicines. But if you can think about using a small molecule as a catalyst for the brain to physically heal itself, now maybe you have to take the medicine once a week, once a month, once a year. That's unclear, probably will depend on the specific molecule and the specific indication, but it's not the same paradigm. It's not the daily administration of something like an SSRI.
So I am incredibly impressed with your synthesis of information and your innovative forward thinking of how you might be able to address this problem, and then your your fearlessness at jumping right into the commercial world and developing a company. So why don't we have a video that I think we're going to show about your company. It's very short, and then I'm going to ask some questions about how you as an academic faculty member or student or trainee address this brave new world that you're doing very successfully. So let's watch that video if we can.
The prefrontal cortex, or PFC, is a brain region that controls mood, cognition, joy, fear, and more. It helps make us who we are. In many neuropsychiatric diseases, neurons in the PFC atrophy or wither. Branches retract, synapses are lost, leading to impaired cognition, disrupted mood, and other symptoms. Psychoplastogens are molecules that spark regrowth of key neurons, healing damaged neural circuits. By physically changing the structures of neurons in the PFC, psychoplastogens hold the promise of rewiring the brain to heal the mind.
It's a fantastic video, so please visit their website and take a look at that. It's very inspirational. So in thinking about your discoveries in your lab and how you can translate that into developing a company, can you tell me what inspired you to do this, and then how you found each other and how this company got launched?
Honestly, I wouldn't say I was inspired to start a company. I would say that I feel a moral obligation as an academic that if we make a discovery that we think can really help people, we owe it to the world to try to bring that to patients so that people can actually benefit from it. And so that's the real reason I started a company. I got into science to be an academic. I'm not into business really. And so I guess that's why I tried to do this. And the way I was able to do it was by surrounding myself with people that are much smarter than me. And I think it's really important to know what you don't know. And for me, I felt like, on the scientific side of things, we have a really interesting idea, but to really make it successful, to do what Mark was talking about and translate this, to make it commercially viable, I had to really enlist the help of other people that understood that world better than I did. And so to do that, I relied on a bunch of resources. So I was very fortunate early on in my career to get one of these STARE grants from Venture Catalyst. And anyone else in the room that are applying for this, I'd really highly encourage you to apply for this program. It's great. As part of that, I went to an entrepreneurship academy. That really helped me to start building my network, to start talking to people and to see what does it take to build a company? What are patients and payers eventually going to be looking for? How can I convince other people to get on board and to help me with this? And I would say I started that process maybe back in 2016. And it really wasn't until I had talked to hundreds of people really looking for a good business partner that could help me achieve these goals. And then I met my co-founder, Nick Haft. And Nick had a very similar vision as I did. It was clear that he was quite bright and very well connected. And that's when we decided to ultimately start the company.
It's really, really fascinating, and I think inspirational to a lot of the trainees here who were thinking in a more entrepreneurship approach to research. So, Mark, from a company's perspective, how does industry see opportunities like this to partner with researchers at a university?
So companies, whether they're at the scale of Shire, when I was leading a team there of about 2,000, we had about $3 billion in annual turnover in sales, and a mix of other offices and presence around the world. From that scale, all the way to the VC world, where I was after, all the way through to small startup companies like Delix now that are growing quickly. In all three cases, there is someone there who's scouring the academic landscape at any point, or working with third parties who specialize in understanding and taking snapshots of the landscape in whatever particular field of therapeutic interest they have, I would say on an annual basis. So the first thing you need to understand is there are people out there looking all the time. So that's the first piece of good news. The second piece is that the fragmentation of in-house R&D at vertically integrated pharma companies into more disparate, opportunistic, horizontally integrated search and development plays in biotech, VC, and elsewhere has really like ossified over the last 10 years in a good way. So it's a long way of saying people like acquiring innovation, not trying to build it all in-house. So there is a large appetite. The challenge I'd say everyone needs to be cognizant about is that there can be a perception or misperception that it can be difficult to work on some occasions with tech transfer offices. And in my experience, I can't speak highly enough about UC Davis, the experience I've had here, and never had any prior to working with David and the team here, but had good corroboration from some other colleagues of mine who'd done some work in the past with UC Davis as well. So you guys should know that at least in the N of one, there was a good reputation out there. But there are other faculties, there are other universities that I've worked with in the past that if someone asked me today, would you work with them? I'd say, I'd think twice. I think, is this really, really worth it because of some of the challenges inherent in partnering with them? And I think the simplest way to distill that to you and how to think about things, if you are interested in building a company, if you are interested in scaling to grow, is think about any relationship you have, any licensing deal you make, any partnership whatsoever as a starting line and not a finishing line, okay? What I mean by that is in cases that I've seen or have seen in others where there may have been challenges of partnering with universities, it's where an agreement, a license, a tech transfer, whatever it may be, was viewed as a final, okay, great, we did it, we made the deal, now you go off, company, you go and make something of it and check the agreement every time you need to see what to do when you have to cross the bridge on a decision, as opposed to the companies, as opposed to the teams who truly view it as a long-term relationship, who view it as the beginning of a journey together in research, in development, in relationship building, and reputation building, and all those things, and truly view it as the beginning of a long-term relationship. I hope that makes sense, but that's the simplest way I can describe the difference I've seen in the past. What I also want to, I can't overstate, is in all you heard from David's example of how he formulated and brought forward the concept that eventually became Delix, there was a lot of self-starting in that. There was a lot, that entrepreneurship boot camp, the relationships he built. Correct me if I'm wrong, David, but no one like came to you one day and was like, hey, you should go to this, you should do this, you independently thought of this, you took a step forward and you did it. And in my life at least, the times in which I've stretched to do something, or more importantly, didn't stretch to do something, were cases where I felt something seemed unattainable, right? Like, oh, I can't learn that particular skill, or I can't learn how to do that. That's for those people out there. They have something different than me. I don't know how to acquire, like, that just doesn't seem like something I could do. And the truth is, we all can do all these things, but there's no substitute for the hard work and the relationship building necessary to get there. And that, quite frankly, can take a while. It can be hard work. It's extra, but I don't want to overstate the amount of hard work that David has done and will continue, has continued to do to help bring this to fruition. So starting line, not finishing line.
Yeah, and I just want to emphasize something that Mark said. Starting a company is just the first step in the journey. And if you want to make this company successful, you got to be present and contributing every step of the way.
I'll also just add one thing there, ego, or how the humility. So, David spoke about surrounding himself with people who had skills or had done things that he didn't have, and assuming you can't know or don't know everything. That's no different for me. It's no different for anyone on our team. And that, I think, is one of the secrets of Delix's success so far. We've been very fortunate to be able to attract some of the best and brightest talent who are all share this long-term mission, this long-term vision of doing something different in mental health, to actually bring a healing-based approach to a lot of these neuropsychiatric and eventually neurodegenerative conditions over time.
This is terrific. I have a lot more questions on here, but we're running out of time. I know that we all wish you both incredible success because we will all benefit from the new therapeutics that you develop. So thank you so much for your hard work.
Thank you.
So I'd like to thank you all for coming today. This has been a terrific turnout for the first Research Expo in a couple of years. There's a lot more to see today. Our speakers will be out at the lunch, and we hope that we'll get to talk to you there. Thanks for coming.